Great. All right, let's get started. Today, I'm going to talk about just generally about drug development. And then next day, we'll get into how machine learning and deep learning are being used to aid and improve drug development. But I think it's helpful to really get an overview of the whole process um, and just really get an understanding for how a drug goes from an idea to a product on the shelf. Okay, so what is a drug? Um, in technical terms, it's a substance recognized by an official formulary or pharmacopoeia. So it's a substance with a detailed prescription on how it can be made and purified and things like that. It's intended for use in the diagnosis, cure, mitigation, treatment, or prevention of a disease. It's something different than a food. There is a regulatory entity called medical foods that are treated differently than drugs. Um, and it's something that's different from a medical device. So this includes both small molecule drugs, and that's mostly what we're going to focus on in this section of the class. But it also includes biologics, and like a lot of our human beings, pretty much every culture has a history of using medicines, using drugs. So using drugs is something that's just fundamentally part of being a human. And that means we've been discovering drugs for a very long time. In fact, some of the drugs that we use today were just initially discovered as extracts from natural products like plants. So the poppy for pain relief, or willow tree extract, um, uh, artis, Artemisia was used to treat fever, and today we know these drugs as opiates, aspirin, and artemisinin. But now we have a different framework for drug discovery and drug development, and it's quite complex. And the modern framework really goes from a basic science idea about a molecular pathway that we want to interact with, um, all the way to a molecule or maybe a protein or natural product that has some effect on that pathway. Um, and we'll talk about both the scientific aspects as well as some of the regulatory and business aspects that kind of shape this process. So the way that we go about drug discovery today is quite different. It's more intentional. So in general, there's a disease target, and we learn something about its molecular underpinnings, and then we start an intentional and broad search for therapeutic agents. That involves our understanding of the disease biology and the molecular mechanism, and it's very interdisciplinary. So, we need biologists, chemists, engineers, you guys, bioinformaticians, computational biologists, all the way to people on the business side and the legal side. And bringing a drug to market typically is an endeavor that requires thousands of people, and can cost, on the order of $1 billion just for one drug. And part of the reason it's so difficult is the FDA has set a very high standard for proving both the efficacy of the drug as well as its safety. And so it's an expensive process. And so it's not one to kind of be undergone lightly. So the business model is that companies are incentivized for innovating these therapeutic solutions, but that also means that the investment is uh, depends a lot on the business model as well as the therapeutic need. So, we'd like to think those two things are always aligned, but sometimes the greatest unmet medical need is not necessarily the one that is the ideal business model. And so a key example of this might be antibiotics. There have been very few new antibiotics approved in the past few decades, even though we know that antimicrobial resistance is growing. But there are some key problems with the business model. So if you were to invent a new antibiotic tomorrow that really overcame an emerging antimicrobial resistance, probably the best thing to do with that drug is to lock it up, put it in a safe, make sure nobody uses it so that we can save it until we really need it for the most serious cases, and for the most resistant organisms, because we don't want the drug out there in the population. We don't want the microbes gaining immunity to that drug. So you can see already there's that's a case where the business model is not really aligned with the therapeutic need, because if you lock it up and that means you're not selling it, and so there's less incentive for pharma companies to pursue new antibiotics. And a lot of people are working on entirely different models for antibiotics. For example, people are working on a subscription-based service, right? So you have a subscription. So in the cases of greatest need, you can use the drug, and in the meantime, you pay a subscription to have access to that. Okay, so we talked about what a drug is. What kinds of drugs are there? Well, there's basically two broad classes. One is small molecule drugs, and the other is higher molecular weight biologics, and biologics are derived from some biological source. I'll mention two, and there are more special classes within biologics that are actually quite different than the standard protein-based biologic, and that's live biotherapeutic agents. That's a space where I've done a lot of work. So that's like microbiome-based therapies using living organisms as a drug. And then of course there's cell-based therapies, and that's generally like human cell therapies. So each of these different types of drugs has unique characteristics, unique manufacturing, and also their own idiosyncrasies in the regulatory pathways. And there are other types too. So phage therapy is one exciting kind of emerging type of drug that would be a biologic and is not necessarily doesn't neatly fall into any of these categories. And vaccines would be another, a similar example. Okay, so the category that we're going to talk about primarily for this section of the class is small molecule drugs. So these are generally lower molecular weight compounds, usually less than about, let's say, 500 Daltons. And these can be, generally, they're synthesized chemically. And they don't necessarily, they might start as a molecule that's extracted from a natural product, but then they're usually optimized to something that's produced synthetically. Typically with small molecule drugs, the administration is oral, although it can be through other routes as well in specific cases. And these small molecules, generally, we know the protein target that they're bound to, and so a lot of modeling goes in to figure out how to really optimize them to a particular binding site on one particular protein. And in general, they should be capable of diffusing across cell membranes in order to be effective. So examples would be probably most of the drugs you're familiar with: aspirin, metformin, statins. Um, Generally, these drugs can be made into an oral formulation, and oftentimes they're just freeze dried into a powder, and we press them into a pill, and they sit in your they sit on the shelf of the pharmacy, and they sit in your medicine cabinet at home, and they don't need refrigeration, and so the distribution is a lot easier than some of the other drugs. Um, Biologics are kind of the other main class. And these typically a biologic is going to be a protein-based drug. So, for example, like an antibody. Uh, those are more kind of recent inventions, although now the pathway to bring antibody biologic to market is kind of well trodden, and many of the new drugs coming out, I think most are actually biologics. But biologics are really any drug produced through biological processes, so fermentation or cell culture. They tend to be proteins, so much larger than the small molecule drugs. And generally, you don't eat biologics. They're not administered orally. It's typically through injection or IV. Because they're proteins that are generally binding to other proteins, they're highly specific. So an example of this might be Herceptin for breast cancer, or insulin for diabetes. So the advantages, highly specific, like we talked about, and because they're specifically binding to one protein target, they're generally they might not have some of the toxicity that's associated with some of the small molecules, because they're protein based, so they can be degraded into fairly inert compounds, as opposed to some of the small molecule drugs, where the process of metabolizing them can produce toxic intermediates. Disadvantages, kind of the opposite of the small molecules. These often have to be refrigerated, and so they're and they have to be extracted and purified from cells. So they're difficult to manufacture, and they're difficult to distribute, and also difficult to administer. For example, if it needs an IV or something like that, then you're going to have to go to a doctor to get it, rather than be able to administer yourself in your own home. Live biotherapeutic agents are kind of a subclass of biologics, and rather than proteins, in this case, I think this is an exciting emerging field that I've been had the opportunity to be part of. And so this is drugs that are actually living organisms, like bacteria, parts of the microbiome. So if we look at a particular disease and we characterize the bacteria, let's say the gut bacteria of patients that have the disease, and compare that to patients who don't have the disease, we might notice, oh, there's specific bacteria that seem to be present in the healthy patients that are absent from the sick patients, right? And now that's a hypothesis for a potential drug. So if we can culture that bacterium, grow it up, put it in a pill, feed it to patients, maybe that will have some impact on disease. So that's a way that we're able to go from DNA sequencing, which we're very good at, to directly formulating hypotheses for a drug. So that's one of the reasons I think microbiome-based therapeutics are quite interesting. They sort of bypass a lot of the workflow that we're going to talk about today, which is takes a long time, takes a lot of money. They can kind of bypass that completely, And another advantage of using, let's say, components of the human microbiome is if these are bacteria that are generally found in healthy people, then the probability of them having strong toxicity is probably very low because it's sort of a normal part of the human flora. So those are some of the advantages. One of the key disadvantages is it's very difficult to regulate and standardize. And so a lot of the terminology that we'll talk about that's part of the drug development process, it's hard to figure out how these agents fit into that. So, for example, if we talk about the dosing of a microorganism, if that microorganism is going to thrive and colonize your gut, it's going to be there maybe indefinitely. It could be there for years or maybe for the rest of your life. So that really creates a lot of additional questions. And when we dose these organisms, if they're actually growing and colonizing the human body and then subsequently growing, then there's a disconnect between the dose and the administered dose and the effective dose, right? You might administer only a few cells, and then you end up with half a kilogram of biomass like growing in that patient. So dosing becomes very difficult. So it's an exciting emerging field. As far as I know, I don't know that there are any components of the human microbiome that are currently FDA approved for use as a therapeutic. Although I think they will be coming. There are many people working on them. Fecal microbiota transplantation is sort of maybe the closest thing we have. So patients who get infected with Clostridium difficile, kind of the most common hospital-acquired gut infection, are often, if they fail antibiotic therapy, they're often treated with fecal transplant. Cell therapies are very exciting. So this is where we use live cells, generally human cells, that are introduced into a patient. These can be the patient's own cells or cells from another individual. These generally have to be administered intravenously or directly injected into the into the site of action. So this includes things that you've heard about, like CAR T cell therapy for fighting cancer, as well as stem cell therapy. So the big advantage here is these can be highly personalized and have a high specificity for let's say targeting cancer cells. And the disadvantage is very similar to the live biotherapeutic agents. We're just getting a handle on how to manufacture these, how to regulate these. But it's another kind of exciting emerging subfield of biologics. And I want to mention, in addition to these types of drugs, I think one of the things, yeah. Oh, yeah. What's that? Why do phage have to be refrigerated for phage therapy? I don't know if they do. I mean, many biologics do, but I don't know phage in particular. Yeah, sorry if I made that claim without realizing it. So companion diagnostics are, I think, a really exciting field, especially for you guys to be aware of. So it's not a type of drug, but it's a way to really make better use of the drugs that we have, and this is another way that we can kind of take advantage of precision or personalized medicine. And so typically in a drug trial, if you look at the diagram under conventional therapy, this is what we expect for a drug, right? we have a population of patients. And for some diseases, maybe the disease is, it's a genetic disease. It's related to like one specific allele, and it's very homogeneous. Okay. Then we might expect that a drug that directly binds to that target is going to have a fairly homogeneous effect, across that cohort of patients. But then there are other diseases where we don't really understand fully the molecular basis and the genetic basis of the disease. So something like IBS, irritable bowel syndrome. It's really a collection of symptoms, and there are drugs that are effective, but in some sense, it's probably not one disease. It's a group of individuals who are kind of clustered together based on having similar symptoms. So if we have a drug for a very heterogeneous disease, a heterogeneous patient population, we might have a very effective drug. And so for a subset of those patients, we might have a drug that, has close to 90% efficacy in curing disease. But that subset of patients might only be 10% of the population. And so then we run a drug trial. We get a subpopulation that, where, it's very effective, a subpopulation where it's basically inert, nothing happens. And then maybe another population that has severe adverse reactions to the drug. And when you look at the trial overall, it's probably a failure as a marketable drug. And the idea is that if we have diagnostics, because now we can collect a lot of data on patients, we can look at gene expression, we can look at the genome sequence, we can look at serum metabolites. We have a lot of ways to characterize the patient population. With that data, maybe we can learn a priori before we treat a patient, what is their response to this drug going to be? And so this has a lot of promise, maybe even to revive some drugs that have failed earlier clinical trials by identifying the subpopulation of patients where the drug is actually effective. Okay, so there's a few examples of this. One of the probably most widely known is HER2 testing for breast cancer, whether it's positive or negative. That's going to impact what drugs are used to treat that cancer in the clinic. Same for PD-L1 expression and immunotherapy. So this is also in kind of an exciting new field. Okay, so I want to talk about two aspects of drug development. So often you'll hear PK/PD as a term in drug development. That's pharmacokinetics and pharmacodynamics. Pharmacokinetics we can think of as what does the body do to drugs once we ingest them or inject them, administer them somehow. Pharmacodynamics is what do drugs do to the body? Okay, so we can start with pharmacokinetics. This is really one of the first things that we look at because it's important for understanding the dosing of the drug and how frequently it's administered, and also the potential side effects. So the fundamental concepts of dose and response, absorption, distribution, metabolism, and excretion all fall within kind of the umbrella of pharmacokinetics. So absorption is really, how do drugs enter the bloodstream or the site of action? Distribution is how are they dispersed throughout the body? So, for example, some drugs might need to cross the blood-brain barrier to have an effect. So the distribution of whether they're able to do that or not is important. because they could be at high concentration in the blood, but if they never cross the blood-brain barrier, they can't have an effect on the central nervous system. Drugs generally have a half-life in the body, and so we want to understand and characterize how they're metabolized, so how they're degraded and potentially broken down into intermediates or even conjugated to and chemically modified in some way. And then how, once they've been processed, how are they eliminated from the body, and what's the timeline there? Because that's going to affect the dosing regimen. So absorption is, we'll think about it as movement of the drug from the site of administration, that could be oral or, directly injected. And that route of administration can have a big impact on the absorption. The drug itself, how hydrophilic, hydrophobic it is going to affect things, as well as blood flow and pH at the site of absorption. And it's going to really influence how much of that drug is bioavailable, right? You can ingest a lot of a compound, and if it's not being absorbed, then a lot of it's not not really available to carry out its therapeutic effect. So distribution is the dispersion of the drug into different compartments in the body. And obviously blood flow to different tissues and organs is important here. Many drugs are bound up by proteins that are circulating in the blood. And as we mentioned before, a big one is the blood-brain barrier. And one of the terms that you'll hear is kind of the volume of distribution, and it's really a measure of how extensively a drug spreads in the body. So you can start out with an injection. Let's say all your drug is in the bloodstream, but then it may accumulate in different tissues throughout the body. Okay, it's worth spending a few minutes on the blood-brain barrier. And this is like a highly selective semi-permeable barrier that separates the blood from the brain's extracellular fluid. So there's a epithelial layer between the brain and the bloodstream that has very impermeable tight junctions between those cells that are tighter than you would see in normal tissues. And that epithelial layer has a number of very specific transporters. So it's relatively impermeable, especially to hydrophilic compounds, and especially to very large compounds. And many of our drugs are both hydrophilic and or large, and so it's difficult to get them across the blood-brain barrier. And in cases of metabolites that are needed in the brain, there's a series of transporters that bind very specifically and pump those across, right? And that's a way for the brain, kind of the most important organ in the body, to sort of isolate itself from changes in the chemistry of the blood that might not have as much of an impact on other tissues in the body, but could be very damaging for the brain. So this creates a lot of challenges for drug delivery, and there's a number of different strategies to overcome these. One is design drugs that are more likely to cross that barrier. And so that's kind of one aspect where we can think about, are there computational ways to predict which molecules are most likely to cross? And then some emerging methods are using nanoparticles. And often you'll see drugs with intranasal administration. And that's sort of it's sort of a shortcut because then you can put drugs directly on the olfactory nerve or the trigeminal nerve through the nasal cavity, and you actually avoid that epithelium. You're just applying the drug directly to the nerve cells, and then it can make it to the brain. It's a relatively small route. So if you require a very high concentration of your drug in the brain to be efficacious, it's very difficult to get that through that intranasal route. So it's really only useful for very potent drugs. Okay. The next aspect of pharmacokinetics is metabolism. So we ingest these drugs, and they generally don't stay in the same form that we ingest them throughout their entire life cycle. In fact, many of them get metabolized in order to convert to their active form, right? Some of our drugs are inactive when we ingest them and then later become active, maybe reacting to the pH of the GI tract or the blood, or maybe passing through the liver, things like that. There's generally two phases of metabolism. And so the first is modification. And here the cytochrome P450s are quite important. There's a few dozen of these in our body. They're a few different homologous gene families. And there's many pseudogenes. So these are like olfactory receptors. They're enzymes that can act on and detoxify foreign chemicals. And because there are so many pseudogenes in the human genome, it seems likely that we've encountered many chemicals in the past that we've needed to detoxify, and these enzymes have kind of specialized in different types of chemistry. And then for drugs that are very hydrophobic, it's difficult to get them to enter into the excretion system. And so for those, what the liver will do is conjugate them with something that will make them a little more soluble so they can be easily excreted. So I mentioned, I think, last day about my prior work doing wastewater surveillance for COVID. We also have done a lot of wastewater surveillance for different metabolites, including drugs. And so one of the interesting things that you see is if you look for something like ibuprofen, you're generally not going to see it. You're not going to see it at least very upstream in the wastewater system near people's homes. What you will see is an ibuprofen that has a glucuronide attached to it, right? So the liver attaches these things, then they can get peed out usually, and then that's what we detect. And what's interesting there is then you can look for chemicals in the environment. It might be in the wastewater, it might be somewhere else, and you can tell which chemicals have actually been processed by the human body, or by some animal. And so part of this process, is important for breaking these chemicals down, but it can also create toxic intermediates. So if we look at acetaminophen, so like Tylenol, that generally gets broken down by a cytochrome P450. There's a specific cytochrome P450 that metabolizes it into a fairly toxic intermediate. And so if you have a lot of that other cytochrome P450, which is usually expressed at lower levels, but if you have a lot of it, then it can take your acetaminophen and instead of breaking it down into kind of inert compounds, it will break it down into highly toxic compounds. And so the cytochrome P450 that creates a toxic byproduct, it's also the cytochrome P450 that gets upregulated if you're a chronic alcohol drinker, for example. So if you are ingesting a lot of alcohol, your body's going to upregulate that gene. So if you're consistently drinking alcohol and you take Tylenol, if you look at the warning on the package, it says if you have, I don't know, 4 or more drinks per day, like you should not take this medicine because of liver damage. That's because there's these competing pathways for how it's broken down, and it can be changed by like what else is entering the body. So we talk about glucuronidation helps these chemicals get eliminated from the body. So this is primarily through the kidneys or the liver, but they can also be eliminated through sweat, through the lungs, through breast milk, and that's why we do extensive testing on women who are either pregnant or breastfeeding, because these drugs and their byproducts can actually be in fairly high concentration in breast milk and might have an effect on growing children. Okay, and so excretion has a big impact on the half-life of a disease, and so there's a few numbers that are kind of important in understanding the concentration over time of any particular drug. And so, we can imagine that if a drug is taken, there's going to be some period of time where that drug, maybe it starts out in one compartment, let's say the gut, if it's an orally ingested molecule, and then it's going to make its way into the bloodstream. And so it's going to, decay kind of, we imagine like a exponential decay from the gut into the bloodstream. And so in the bloodstream, we see it going up. And then based on however much is in the bloodstream, now it's going to start to be metabolized and excreted. And so we'll see it kind of leave that compartment. So this would be, generally we're monitoring the level of drug in the blood. And so for a particular dose, we can calculate the Cmax, and that's the highest concentration of the drug in blood. It might be significantly lower than the amount of drug in the pill due to absorption. We can calculate the Cmin. So if it's a completely synthetic drug that doesn't have any natural analog, the Cmin after some washout period is probably 0. But if we're talking about a drug, like insulin or something like that, that's naturally produced by the human body, then the Cmin is not necessarily going to be 0. And the other important characteristic of this curve is the half-life, right? And so that's going to generally determine the dosing schedule of a drug. And so if it's a drug we need to be active all the time, and it has a half-life of 4 hours in the body, then these are pills that we're probably going to have to ask patients to take like 3-4 times a day. Whereas if the half-life is a week, then then it's a entirely different dosing regimen. Interestingly, like most pills, we want patients to take every day, because any other dosing regime, patients tend to like fall off of and not comply to. So it is much harder to get a patient to reliably take one pill every other day than it is to, just take half that dose every day. And certainly like the once every 3 days, once a week are some of the worst. Okay. So how do we model the pharmacokinetics? Oftentimes it's just really just like a one compartment model. So if we look at the top, A0, that's just the initial bolus of drug. Maybe it's a pill and it's into the gut. A1 would be the blood. And then it's just an exponential decay from A1 into A2. A2 would be excreted. But we can think about more interesting models, like the two-compartment model, where it goes from the gut to the blood, to some peripheral compartment, and then has some equilibrium there, and then gets the fraction of the drug that's in the blood, is open for excretion and processing that way. So generally, this is how we model it. If it's an injected drug, then we can skip the GI tract altogether, and often just approximate it as the entire bolus of drug is just sort of instantaneously in the bloodstream. And then the one compartment model becomes very simple, right? It's just a single exponential decay. Okay. Okay, so all of these pharmacokinetic factors have an impact on toxicity. So if we have a poor absorption, then we have a low therapeutic effect, and we kind of need more of the drug, which might create more side effects. The drugs can accumulate in different tissues. So lipophilic drugs might start to accumulate in the fat or in the liver. We talked about production of toxic metabolites like acetaminophen. And then especially in patients who might be complicated patients and have some kidney or liver problems, then excretion can break down, and that can also be a cause of toxicity. Okay, so that's kind of an overview of pharmacokinetics. Pharmacodynamics is, generally, like, what do these drugs do to the body? So we'll talk about drug receptor interactions, dose response curves, mechanism of action, and how they affect in a therapeutic effect or a toxic effect. So oftentimes with small molecule drugs, we're talking about drugs that bind to specific receptors, and in general, these receptors are proteins. So those could be proteins involved in signaling pathways, or they could be enzymes. And one large and very druggable and exciting class of protein are the G protein coupled receptors. We also have a lot of drugs for, like I said, enzymes or channels. The class of drugs that activate their downstream target, we tend to refer to as agonists, and the drugs that inhibit their target are antagonists. But we can also have partial agonists who might bind to a receptor, but they don't stimulate the receptor to the same degree as the natural ligand. The other term that's worth talking about here is affinity. So that's simply the strength of the drug receptor interaction, and so we can model that as a concentration where the receptor is 50% of the receptor is bound. Okay, a dose response relationship is really how much of a phenotype do we see for a particular dose of the drug, and the dose that produces half of the effect that the maximum effect is the EC50. The Emax is the maximum effect regardless of the dose. At some point with most drugs, all the receptors, 100% of the receptors are bound, and so putting more drug in, it might increase the side effects and the toxicity, but it's not really going to in fact impact the pathway that it's targeting. And the therapeutic window is related to this, and it's really like the range of doses that can have an effect but avoid toxicity, and we'll talk more about that. So here's an example of a dose response curve. I took this from Wikipedia under because I Googled dose response curve. And I like it didn't say what drug this is, but it looks like a terrible drug because the EC50 is almost 1 molar, right? And so for good drugs, we're looking at something like one nanomolar. So this is a bad drug, but it kind of illustrates the concept that we're talking about. Like above that EC50, um, you don't get once you go one more log unit up, you're not getting any additional efficacy out. Okay. So with PD or pharmacodynamics, we often talk about mechanism of action. And so if you ever are, your doctor says, oh, you should take this drug or that drug, and you're like, oh, how does it work? If go to the literature or even go to Google and say, what is the MOA of this drug, it'll generally give you a good description of what the drug is doing at a molecular level. So if you take the example of beta blockers, bind to different beta blockers bind to different subsets of the adrenergic receptors, generally the beta adrenergic receptors. And like those are probably like most densely populated in the heart, and so these are good heart drugs. They'll tend to lower that fight or flight response pathway. They'll tend to slow the heart, reduce the strength of the contractions, things like that. And so this is useful for a lot of indications in cardiovascular medicine. These things they can have one intended mechanism of action, but they can also bind to a lot of other receptors. So with the beta adrenergic receptors, some of them are very specific for one subset of those receptors. Some of them bind many, and even the alpha adrenergic receptors. So the specificity is going to have a lot to do with the side effects and the therapeutic effect. Okay. We mentioned like a lot of these, some of these drugs are let's say binding to enzymes, and in that case, the action is pretty straightforward. It's just generally blocking the activity of that enzyme. But many of them are binding to receptors, and that means that there are many steps between the drug binding its receptor and the phenotype downstream. And so understanding these signal transduction pathways is important to predicting the effect of these drugs, because they can feed into signaling pathways where there's other inputs too, and it can make them dependent on a lot of other factors besides just the particular drug that you're administering. It's worth talking about tolerance and sensitization. So we're all familiar with tolerance. So after you've taken a particular drug for a while, the body often will respond either by generally ramping up metabolism to break down the drug, or even down regulating the target of that drug. And that can affect the dose required to kind of have the same effect. And so we can get desensitization on kind of a shorter time scale of the receptor due to repeated exposure. That's often an intrinsic part of different signal transduction pathways. We can get over longer periods of time downregulation. So, the different cell types might downregulate the total number of receptors for the drug to bind to. And then in some cases, the more you use a drug, the stronger the response. And so you can get sensitization as well. Toxicology is the study of what are the negative side effects of a particular drug that might have some other therapeutic value. And so there's a number of different types of toxicology studies that we do. Can look for acute, just reactions to the drug. Look for DNA damage, effects on reproductive health, as well as many compounds are carcinogenic, and so some of those studies are very costly to perform because they require kind of long time scales. And in general, in terms of evaluating the safety of a drug, we also want to look at what are the general effects of this drug on vital organ systems like the central nervous system, the respiratory system, cardiovascular system, and these toxicology studies are often done in animal models first. The therapeutic index is an important concept in kind of understanding like the range of dosing for a drug. So ideally, you want quite a large therapeutic index, and the index is really it's the distance between the dose of the drug that is toxic or maybe lethal, and the dose that starts to have a therapeutic effect. And so we measure it from the 50%, either 50% of the time we see a toxic response, or 50% of the time we see an efficacious response. And if those two are very close, then it becomes really challenging to dose that drug. And so I think like warfarin is one example. It's a blood thinner, and at too low a dose, basically has no effect. At too high a dose, it's dangerous, and the distance between the effective dose and the toxic dose are quite close. And a number of papers kind of in my field of microbiome have pointed to the microbiome metabolizing warfarin in the gut as being adding some variability to this. So it makes it even harder to dial that dose in, because you don't know how each particular person is going to respond. Okay, so that makes that narrow therapeutic window makes for a more risky drug. Okay, and drugs can interact with each other. So in synergism, like alcohol and sedatives, you might get a mild effect from a particular dose of each, but when you combine them, it can be quite risky. Drugs can antagonize each other. So this is a common situation where they have maybe differing effects on the same downstream pathway, or maybe one even occludes the other from the binding site. And that an example might be naloxone would occlude opiates from the opiate receptors, and that's how it acts. And then you can have things like in general anti-hypertension drugs lower blood pressure, and then you can put two together, and generally the effects are additive. So then it's easy to kind of calculate what the combined dosing is. Okay. All right, so we talked a lot about the drug development process and what are the different ways that we characterize PK and PD. So I want to spend the rest of the time talking about what's the process of finding these drugs. So it's a multi-step process. We're going to start by identifying a target, and this is generally a protein in the body. We need to validate the target. Then we need to identify hits. So this will be like a large number of compounds that we think, have a chance of interacting with this target in the way that we want, either as an agonist or an antagonist. There's a stage called hit to lead, and that's where we take these chemical hits and we say, okay, let's spend a little bit of time and effort developing them further and try to get the best chemical structure that is similar to the initial hit before we pass it into the lead pipeline. And so we're going to, at the level of lead identification, we're going to take the best looking chemicals that came out of the previous stage, and then we're going to try to optimize them, and then measure all these properties to try to improve how efficacious they are, how safe they are, try to get ideal pharmacokinetics. And so target identification. This is a key area where deep learning, machine learning can play a role. And the idea here is if we have a disease, what's a good target to hit? Like, what's a good gene in the genome? What's a good protein that we'd like to look at? And in general, we're looking at proteins, enzymes, receptors, but it could be anything. It could be, structural RNA or something like that. And here we're just generally using bioinformatics. So we can use genomics, proteomics. We can use our knowledge of the molecular mechanisms of a particular disease. We can do high throughput screening, let's say looking at knockouts of a gene in a mouse. And then we really have an almost unlimited number of bioinformatic or AI or deep learning based tools that you guys could imagine. And the key considerations here are, how closely connected is this target to disease? If I knock it out, does it clearly, affect some biomarker of disease or disease progression in some model system? And is it druggable? Okay, is this a protein that I can actually make a small molecule drug to agonize or antagonize? And another consideration might be, is there a structure for it? And maybe there is. Maybe there's an alpha-fold structure, but no crystal structure. So what do we mean by druggability? Druggability is just, if we start with this target, and let's say it's a protein, what's the likelihood that it could be modulated by some small molecule to produce a therapeutic effect? So what wouldn't be druggable? Well, proteins that have a lot of disorder, right? Proteins that only adopt a 3D conformation when they're already bound to some other complex, something like that. That's going to be very difficult to try to drug. Protein-protein interfaces tend to be fairly difficult to drug. And things like transcription factors, right? In one of the first classes, we looked at the 3D structure of a transcription factor, and I showed you the part that's fitting into the major groove. It's generally like a big flat surface that's determining the specificity there, and there's not really a pocket in there for the protein to bind to, right? So yeah, we covered kind of most of this. The target expression is also important. So if the target is not expressed in disease tissues, then that's obviously going to affect the druggability. And having protein structures and seeing if there's pockets that like where a particular protein can bind, that's going to be like one of the ways that we can assess druggability. So one of the reasons that we talk about these drugs either binding to enzymes or binding to receptors is that enzymes intrinsically, like, they have an active site, and that's generally a place where a small molecule binds. So those tend to be druggable because we can start with a compound that maybe looks a lot like the compound that they have their natural activity against. And then we can, add to it to make it a even tighter binder, for example. Or if it's if it's a receptor, there's a small molecule ligand that binds there, and we can, begin with a hypothesis for a drug that sort of builds off that small molecule ligand. Okay, so after we identify a good target, let's say it's an enzyme and it's got a nice active site that we think is druggable. Now we want to really validate that it is important in disease, so we can use CRISPR screens, overexpression, use animal models, and then modulate the function of that particular gene. And then in humans, often we want to look at different biomarkers if we can't, biomarkers of disease if we can't, directly just get a faster readout of disease. And this ensures that if we choose a molecule that biochemically does what we want with respect to this target, that we're actually going to induce a therapeutic effect downstream, because we can invent a drug that binds really well to a particular active site, but if we don't have the right target, it's not going to be efficacious. So that brings us to identifying hits. So and there's a number of ways to do this. Probably the classical way is to start with a huge library of different compounds, come up with a very high throughput assay, either a biochemical assay or maybe a cell-based assay, and screen which compounds give us some activity against the target that we're looking for. A newer approach to this is saying, okay, we generally, like, the compound that we end up with at the end of this process is probably not going to be identical to one of the ones in the large libraries. That may be a component of it. So if we were to take fragments, smaller fragments of chemicals, and put them in at very high concentrations, so they're more likely to bind, and then look at what things have activity. So if we find these smaller fragments that have efficacy, let's say at like 1 molar or something like that, then we can try to redesign them to be efficacious at lower concentrations. And of course, another area that we're particularly interested in here, of course, is virtual screening. So can we design algorithms to identify what compounds might, just given the structure of a enzyme, let's say, can we identify computationally what compounds might bind there? Once we have these hits, from among them, we're going to choose lead compounds. And so we want to do some optimization of the best hits and try to really validate them, and also do some studies, some structural activity relationship studies, to understand how does modifying different parts of this chemical, how does it affect things like activity, and even some of the pharmacokinetics. And then once we've done a little work with those hit compounds, the next step is, before we start characterizing them extensively, we have to, narrow that number down. So we might start with a million hits in our high throughput screen. We might have from there, we might have 1,000 hits, and then for leads, we probably want a few dozen leads, something like that. And once we have those leads, then we're doing a lot of chemistry to refine those. Okay. And structure activity relationships are really important in kind of guiding how that refinement happens. So structure activity relationship is a relationship between a drug's chemical structure and its biological activity. Oops. And so we want to identify which parts of the molecule are essential for activity, and we want to modify that chemical to increase its activity, increase its potency, decrease its likelihood of binding to off targets. So a lot of times, we'll have drugs like kinases. And we know that we want to target a kinase downstream, and there are many general compounds that can inhibit kinases, but they do it very broadly, and so those tend not to be selective enough to have the effects we want. We also want to know how changing the structure of this drug changes the toxicity as well, and how it also impacts pharmacokinetics. And so the process is, we start with our lead compounds. We study how that lead compound interacts with its target. So it's great if we have a crystal structure, maybe even a crystal structure of the target bound to the lead compound. Then we can start to make guesses about how modifying that is gonna change the activity. And then we can make different modifications of that molecule and observe empirically how that works. And if we collect enough data, especially with respect to lead compounds in this particular target, then we can start to predict how different changes are going to impact that. So some typical strategies in optimizing these lead compounds are, changing different chemical modifications. If it's got like an alkyl chain, we can vary the length of it, or if it has a linker, we can vary the length of it. Can add or remove rings or double bonds. And typically, if you look at drugs, what you'll notice a lot of times is, gosh, they have a lot of rings, they have a lot of double bonds. We talked about this earlier when we got into protein structure, and I said when things are denatured, that like all those rotatable bonds can be in many different positions. When a protein is folded, or when a drug is bound to a protein, then it can generally only be in one configuration, right? So there's a loss in entropy from if you have a lot of rotatable bonds. There's a loss in entropy from being free in solution to being bound to a protein. And so what we'll tend to do is, if we can put a ring in or a double bond in so that the structure doesn't have as much wiggle room when it's unbound, then we can get rid of that force that's making the molecule prefer to be free in solution. So you'll see that quite a bit. And then I want to point out QSAR methods. And so this is making quantitative predictions about the relationship between the compound structural properties and its biological activity. And obviously that's a good entry point for machine learning, deep learning. Okay. So a lot of the challenges here are, figuring out what are what's the toxicity, what are the off-pathway effects, and also balancing between changes that maybe improve the binding, but make the absorption worse or something like that. So kind of integrating all of these different points together is another challenge. Okay, I'm going to skip through some of these and just give an overview of kind of the whole process. So drug discovery is takes decades, okay? And even just in, once we've gone through the drug discovery phase and we're really just in the drug development phase, putting it into clinical trials, itself like that, it's hard to do that in less than 7 years. It's a fairly complex process, and I think I mentioned earlier, there's usually thousands of people involved in bringing any drug from the discovery phase to a product that's on the shelf. Once we have what we think is a good drug, and we've done all of our preclinical testing, which is usually in cell culture and on animals, then the next step is to apply for an IND. So that's an investigational new drug application, and these are typically hundreds and hundreds of pages, and it's like an enormous amount of work to put one of these together. You send it to the FDA. If they don't reject it within a certain period of time, then you're not approved, but you're okay to like go ahead with your first study. And that's a phase 1 study, so it's a first in human study. That's generally a very small number of patients. It might be kind of multi-phased, and often with increasing doses. And here you just want to make sure that you didn't miss anything with the animal testing. Like putting this drug in a human being isn't gonna like be highly, highly toxic. So you start out with a very, very low dose. Wait a while, you measure all your PK, maybe measure your PD, see if it has an effect. And then you'll start maybe ramping up the dose a little bit to where you think you approximately want it therapeutically. So that's the phase 1 trial. Here it says 20 to 80 patients. It's often even smaller than that. And once you pass your phase 1 trial, and your phase 1 trial is really just for, toxicity, then you go into your phase 2 trial. And the point of the phase 2 trial, it's still mostly about safety. But you can have a few dozen patients, you can have a few hundred patients in your phase 2, and there's a number of things that you're going to test here. One is you've determined that it's generally safe to put these drugs in humans in your phase 1, but you still don't know exactly what the dose should be and what's the dosing regimen. And so your phase 2 is probably going to try a few different regimens, then measure some biomarkers of disease. In general, the idea is the phase 2 is still just about figuring out things like dosing and mostly about safety. You're putting in a larger population. You're going to measure for adverse effects. But you're also, if you're a pharma company that's paying hundreds of millions of dollars for this trial, then you also want to look at efficacy, although you're probably going to be underpowered to prove efficacy to the level that FDA would like to see. But if there's no efficacy, then you're probably going to make the decision, even if FDA allows you to go to phase 3, you're probably going to cut it short if you don't see some sort of efficacy in your phase 2. Phase 3 trial, that's the big trial. Once you pass that, then you can apply to start marketing your drug. You can actually get approved by the FDA. And those tend to be very, very large trials. So hundreds of patients to thousands of patients. And it depends how risky the FDA thinks the long-term side effects might be. And it depends also on how big the market is. So if you're putting a drug into phase 3 clinical trials, and it's going to be the next greatest statin, right? Like the most popular, it's going to be one of the most popular drugs out there. Then they're going to want you to run a really huge trial because there are so many people taking the drug. That kind of increases the likelihood that someone gets an adverse reaction to it. All that goes well, then you apply for your NDA and eventually get approved, and then you can start to market your drug. Often with drugs, then you'll continue to do surveillance and continue to see if it's efficacious. Like for example, in the case of vaccines or something like that, there's a lot of post-marketing data collection that goes on. And most drugs don't make it through this pipeline. So here we can see there's also big differences between different types of drugs. And so you can see the probability of success in going from phase 1 to phase 2 is often quite high, right? More than 50% in all of these cases. But then phase 2 to phase 3, if we look at oncology drugs, so cancer drugs, can actually be quite low, right? And the overall probability from starting our phase 1 to making it through to approval for a cancer drug is only 3%, and overall it's about 14%. So the vast majority of drugs that enter this pipeline don't get approved, and it's quite expensive. And you can see that the success rates in phase 2 are the lowest. Why do you think that is? Yeah. Yeah. Yeah, you're kind of proven to FDA that it's not toxic, but that's where if you're a pharma company, you're really looking for some response in a biomarker or in disease severity or something like that says, yeah, we should invest in a much more expensive phase 3 trial and try to bring it to market. No. Okay. All right. Okay, so it's a little bit about the FDA. The other important regulatory body is the U.S. Pharmacopoeia and the National Formulary, and they're really setting the standards for how a particular drug is defined in terms of its like manufacturing and purity and things like this. The FDA is an organization that's whose mission is to ensure public health by maintaining the safety and efficacy of different drugs, and they currently regulate over 20,000 approved drugs. And they're largely funded not only by the government, but from industry fees. And we can see like a little bit of the breakdown of what are the FDA approved drugs in terms of indications. So like a huge number in oncology, also psychiatry and also infectious disease. If we kind of dive more into infectious disease, we can see there's a lot of antibacterial drugs, and after that antiviral, and then a few for other parasites. There's not that many new approved drugs in any particular year. And of the drugs that are approved, many of these are modifications to an existing drug, not an entirely new mechanism of action or disease target. So in this example, there are 48 new approved drugs in 2019, but only 20 of those were kind of first-in-class drugs. So only 20 are really novel and not kind of an iteration of an existing drug. The cost of bringing these drugs to market is enormous. You can see here in the millions of dollars. And so, the average cost of bringing a drug to market is about $1 billion. And remember that, only 14% of these are ever make it to market. So it's an expensive process. Okay. So I think that's it. Next day we'll talk a little bit about how deep learning and AI and bioinformatics can help improve this process. Go ahead, and
I guess you like, do you usually stop recording before?
oh, I see. Yeah. That's what we did.